Right this very second, millions of invisible, microscopic operators are literally dictating your health, neutralizing natural poisons, and maintaining the fundamental balance of your entire body. Welcome to The Explainer.
So it begs the question, how exactly do you stop a microscopic machine? I mean, it's a massive question, right? We are talking about the absolute highest of stakes here, how tiny little molecules act as this ultimate control system for the incredibly dynamic, non-stop chemical processes that are keeping you alive right now.
Okay, here's our roadmap for today. First, the brakes of biology. Then, reversible versus irreversible inhibition. Next, we'll hit the 4 types of reversible inhibition, followed by measuring the impact, and we'll wrap up with poisons, medicines, and life. Let's dive in.
Part 1. The brakes of biology, controlling the speed of life.
Okay, so what exactly is an enzyme inhibitor? Well, simply put, it's a molecule that binds to an enzyme and completely blocks its activity, preventing it from converting substrates into products. If you think of enzymes as the high-revving engines of your cells driving all these essential chemical reactions, then inhibitors, they're the vital brakes. Because without them, metabolic processes would just run totally out of control. Inhibitors maintain cellular homeostasis. Basically, they make sure the engine runs at the exact speed required for you to survive.
Moving on to Part 2, reversible versus irreversible, the dimmer switch and the sledgehammer.
Generally speaking, an inhibitor can take one of 2 main paths. The first is reversible inhibition, which is temporary. It relies on non-covalent interactions, things like hydrogen bonds. It's exactly like a dimmer switch in your living room. You can turn the light down to fine-tune the vibe, but you can always turn it back up whenever you want. Irreversible inhibition, on the other hand, is like taking a literal sledgehammer to that same light fixture. It forms strong, permanent covalent bonds, effectively destroying the enzyme's ability to ever function again. Now, the differences between these 2 strategies are absolutely crucial, especially for things like drug design. With temporary binding, the reversible kind, the enzyme's activity can ultimately be restored once that inhibitor is removed or flushes out. But with irreversible, covalent bonding, the damage is done. The activity cannot be restored unless the cell actually goes to the trouble of synthesizing a brand new enzyme from scratch. So it's a totally fundamental difference in biochemical strategy. A reversible inhibitor detaches and boom, back to business as usual. An irreversible inhibitor permanently takes that specific enzyme right off the board.
Part 3. 4 types of reversible, the microscopic musical chairs. When the scientist W. Wallace Quinlan categorized these reversible types back in 1963, he revealed a system that honestly plays a lot like a super high-speed game of microscopic musical chairs. First up, you have competitive inhibitors. They literally fight the substrate for the one single chair, the active site. But if you flood the system with enough normal substrate, you can actually outcompete the inhibitor. Second, uncompetitive inhibitors. Now, these are pretty rare. They actually wait and only bind after the substrate is seeded, locking the whole enzyme-substrate complex in place. Third is non-competitive. These bind to a totally different part of the enzyme. They reduce the activity, sure, but they don't stop the substrate from sitting down. And finally, mixed inhibition, where the inhibitor binds whether the chair is empty or occupied, just with varying levels of affinity.
Section 4. Measuring the impact, the math of the microscopic.
So how do we actually measure these invisible battles? Well, biochemists rely on 2 really key kinetic concepts. You can think of Vmax as the ultimate speed limit. It's the absolute maximum rate at which the enzyme can catalyze a reaction. Km, on the other hand, tells us all about attraction, specifically the substrate concentration you need to get to half of that maximal activity. So a lower Km actually means a higher affinity. By carefully tracking exactly how an inhibitor alters these 2 numbers, Vmax and Km, scientists can figure out exactly what kind of microscopic game is being played.
And to actually visualize these changes, researchers use something called a Lineweaver-Burk plot. Now, I know it might look a little intimidating at first glance, but it's incredibly useful. It simply translates those complex kinetic equations into clear straight lines on a graph. By plotting the data this way, the exact point where the lines intersect instantly gives away the inhibitor's true nature, whether it's competitive, non-competitive, or uncompetitive. It is literally the definitive treasure map of microscopic mechanics.
And finally, Part 5, poisons, medicines, and life, the high stakes of inhibition. This brings us to a frankly brilliant concept known as suicide inhibition, and it's perfectly demonstrated by the drug DFMO. It's essentially a molecular trap. The enzyme gets completely tricked into binding the drug instead of its normal target. But then, the enzyme actually catalyzes its own doom. It triggers a reaction that eliminates a fluorine atom, creating a highly reactive molecule that immediately forms an irreversible covalent bond. The enzyme literally destroys itself. And this exact dramatic mechanism is how DFMO so successfully treats African sleeping sickness.
The stakes here, they couldn't possibly be higher. By targeting specific enzymes, we can create incredible life-saving medicines, like methotrexate for chemotherapy, or ritonavir, which is a really crucial antiviral that blocks HIV's protease. But nature uses this exact same biochemical math for warfare. Nightshade plants, for instance, produce glycoalkaloids, these natural inhibitors that act as deadly neurotoxins just to paralyze predators. Or take paclitaxel, a wildly potent poison derived from the Pacific yew tree.
It highlights a really profound reality about biochemistry. As this quote lays out, many poisons produced by animals or plants are enzyme inhibitors. And conversely, many drug molecules are enzyme inhibitors that target an aberrant human enzyme or one critical for a pathogen's survival. It really is a biochemical tightrope, constantly balancing survival and destruction on a molecular level. So, knowing that a single tiny microscopic connection can either paralyze a predator in the wild or cure a devastating disease in a hospital bed, how are we going to harness the power of enzyme inhibitors next? The future of biochemistry, pharmacology, and honestly human health as a whole depends entirely on how we answer that